Welcome to Bosker Sciences. Today's topic is cell transport. There are four main topics that are going to be covered, which are going to be passive versus active transport, types of solutions, water potential, and types of active transport. You might have the question, what is transport in a cell? And that's a one-sentence answer, which is the movement of substances into and out of the cell. But that's not going to be enough, so we're going to go in depth in our video today. The first type of transport that we're going to talk about is passive transport, which doesn't use any energy. Passive transport can be described as the movement of molecules down a concentration gradient from a region of high concentration to a region of low concentration until equilibrium is reached. Two examples of passive transport that you should know are diffusion and osmosis. Diffusion is the movement of molecules down a concentration gradient, and there are two types of it, simple and facilitated, which both do not use energy. If you look at the diagram on the left, you can see that diffusion just uses the phospholipid bilayer that we talked about in the previous video to go through the membrane, while facilitated diffusion uses special proteins that are hydrophilic to move out of the cell. So let's talk about simple versus facilitated diffusion. As we talked in the previous slide, we said that simple diffusion does not use protein channels, and a couple examples are in the kidneys and countercurrent exchange. There's a part in the kidney called the glomerulus, and that's where solutes dissolve into the blood and they diffuse into the Bowman's capsule of the nephron. And this shows simple diffusion because it goes from a high concentration to a low concentration. Another example of simple diffusion is countercurrent exchange, and I would memorize the definition to this, which is the flow of adjacent fluids in opposite directions that maximizes the rate of simple diffusion. And this is important in fish and other aquatic animals. Basically, blood flows toward the head and the gills, while water flows over the gills in the opposite direction. And this process maximizes the diffusion of gases and waste between the water and fish. The other type of diffusion is facilitated diffusion, and it requires a hydrophilic protein channel, and an example is an ion transport and aquaporins. Aquaporins are important in biology, and they are special water channel proteins that are found in certain cells, and they facilitate the diffusion of a lot of water across the cell membrane. Before we talk about the different types of solutions, we need to understand some basic vocabulary. The first word is diffusion, and we know that that's the movement of particles from high to low concentration. Osmosis is the diffusion of water across a membrane. And the difference between a solvent and solute is that a solvent is the substance that dissolves, while a solute is the substance that's dissolved. So in a saltwater solution, the solvent would be water, while the solute is salt because it gets dissolved in the water. There's three different types of solutions, which are hypertonic, hypotonic, and isotonic solutions. In a hypertonic solution, the concentration of solute is going to be greater than the concentration of solute in the cell. Therefore, water will move out of the cell because water flows from high concentrations of water to low concentrations of water. As a result, the cell shrinks. In a hypotonic solution, the concentration of solute is going to be less than the concentration of solute in the cell. Therefore, water will flow into the cell, causing the cell to swell or burst. If the cell is a plant cell, the cell wall will prevent the cell from bursting, and the cell will merely become turgid. A turgid is a word that many biologists use, and it means that it's crisp, like celery. However, if a plant loses too much water, it loses its turgor pressure and it falls down. The last type of solution is called an isotonic solution, and that's where water moves in and out at the same rate because the solute concentration and inside the cell and outside the cell is at equilibrium. The diagram on the left is one that you'll see often, and if you look at the tube on the left, you could see that the left part of the tube is a hypotonic solution because there's a smaller number of solute than the right side, which is a hypertonic solution. While you look at the right tube, it's isotonic because it has the same concentration, so water will flow inside and out to the cell, or in this case, the tube. In this diagram, you could see that the isotonic solution has the same number of water moving in and out the cell, while the hypotonic solution has more water coming in than water going out, while in the hypertonic solution, more water leaves than enters. I hope you paid attention because you got a quiz. In this quiz, you're going to tell what type of solution it is and what the flow of water is going to be. There's 5 questions and you should take 2 minutes. So pause the video and try to get the answers. If you said number 1 was hypertonic and water leaves, then you're correct. This is because there's a higher concentration inside the cell, which means water is going to have to leave to match the concentration outside. Number 2 is going to be isotonic because it's going to be the same inside and outside, and the flow of water is going to be in both directions at an even rate. Like number 1, number 3 is also hypertonic because it has a higher concentration inside than outside. Number 4 is hypotonic because it has a lower concentration, which means more water is going to have to come in to match the concentration outside. Number 5 is a trick question because I said there's 85% water instead of concentration of solute. This would mean that the solute concentration inside of the cell is 15%, and 15% is smaller than 20%, so the solution is going to be hypotonic, which means water is going to enter the cell. The next topic is going to be water potential, and scientists describe that as the movement of water. Something that you should know is water potential for pure water is 0, and it never goes above 0. Addition of solutes lower water potential, so in cells, the water potential is going to be negative, and water will move from a higher to lower water potential like diffusion. To use the equation, I suggest watching a Khan Academy video on it because that question has graphs and more statistics than I do. I'll put a link in the description, and I recommend that you should watch it because it's only like a 5-minute video. The other type of transport is active transport, and that is the movement of molecules against a gradient. So it goes from a concentration of low to high. So that's why it requires energy in the form of ATP. Pumps or carriers carry particles across the membrane by active transport. An example of this is the sodium-potassium pump, which pumps sodium and potassium ions across the nerve cell to return the nerve to its resting state. In the diagram on the left, you could see that blue is potassium while red is sodium. So more sodium is going in while less potassium is going out, and that's why it requires ATP as energy. Another example of active transport is the electron transport chain in mitochondria, which consists of proteins that pump protons across the cristae membrane. You can learn more about mitochondria in my previous video, which talks about cell organelles. The last topic of the day is going to be different types of active transport. The first is pinocytosis, which is the uptake of large dissolved particles, and does this by the plasma membrane enclosing it in a vesicle. The second type of active transport is phagocytosis, and that's the engulfing of large particles or small cells by pseudopods. The cell membrane wraps around the particle and closes it into a vacuole. This is the way human white blood cells engulf bacteria, and also the way in which amoeba gain nutrients. The last type of active transport is receptor-mediated endocytosis, which is when extracellular substances bind to the receptors on the cell membrane. But I wouldn't worry about this too much because this rarely happens in biology. So that's the end of today's video on cell transport, and tomorrow's video is going to be on cell communication.